Pseudohyperaldosteronism is a condition that mimics hyperaldosteronism. Causes can be environmental or genetic. Although it implies overproduction of the hormone aldosterone, levels are normally low. But why? Cortisol and aldosterone are steroid hormones produced by the adrenal gland that diffuse from the blood into target cells. They both bind with the same affinity to cytoplasmic mineralocorticoid receptors. However, in aldosterone-specific tissue, such as the kidney, this causes a problem. Cortisol levels are much higher than aldosterone, and unless they are stopped, they would outcompete aldosterone for the receptors. Thankfully though, an enzyme known as 11-beta-HSD2 found in aldosterone-specific tissue converts active cortisol into inactive cortisone. Cortisone cannot bind to these receptors. Glyceretinic acid is an active compound of licorice that may cause pseudohyperaldosteronism when present at high levels. It inhibits 11-beta-HSD2, thereby stopping the conversion of cortisol to cortisone. The ratio of free cortisol to cortisone increases, leading to overactivation of the receptors. The activated complexes migrate to the nucleus and bind to DNA, activating genes coding for various channels. Sodium reabsorption leads to hypernatremia, potassium secretion leads to hypokalemia, and hydrogen secretion results in metabolic alkalosis. Water follows sodium passively, causing increased blood volume and blood pressure. Macula-denser cells of the juxtaglomerular apparatus detect the plasma sodium increase, which suppresses RAS. This leads to low renin and low aldosterone levels. Mineralocorticoid antagonists have been used as a common treatment to normalize blood pressure, whereby they block the receptor sites and prevent aldosterone and cortisol from binding.